on about the introduce themselves, which is what they try to do. But they're going to talk about something pretty different here. They're looking at the ability of the organism to be resistant to specific antibiotics. And they're going to give you some pretty interesting information on that. So,
good afternoon. My name is Abhi Kanchura.
I'm Alessandro. I'm Cameron Weller.
It's a pleasure to be here and talk with you all about our research on investigating how Mycoplasma genitalium adapts to finite or invisible antibiotics through genomic sequencing. Firstly, what is Mycoplasma genitalium? Mycoplasma genitalium, or MG, is a sexually transmitted pathogenic bacteria that infects the human genital tracts. In men, it causes urethritis. In women, it causes cervicitis, endometritis, and pelvic inflammatory disease. MG's incidence and its resistance to antibiotics is increasing in the United States. In fact, it is one of the 3 microorganisms that is added to the CDC's watch list for antibiotic-resistant threats in this country. Also, MG is more prevalent than chlamydia globally. Therefore, this makes MG a significant infectious pathogen that requires serious attention. Currently, the 3 first-line antibiotics for treating MG, which are doxycycline, azithromycin, and moxifloxacin, are ineffective. This table shows the success rate and the problem associated with each antibiotic. Firstly, doxycycline has a success rate between 30 to 40%. However, it has very low effectiveness in treating MG. Secondly, azithromycin has a success rate between 85 to 95%. However, MG is slowly becoming resistant to this drug. Thirdly, moxifloxacin has a similar success rate as azithromycin at 85%. However, consumption of this drug leads to severe side effects such as headaches, numbness, and dizziness. Plus, it's not approved for people below 18 years old. Therefore, new antibiotics is needed to treat MG. A clinical study done in 2020 by Weisenfield and colleagues recruited 233 patients with pelvic inflammatory disease. They divided these patients into 2 groups. Group 1 got the original treatment with ceftriaxone and doxycycline, while Group 2 had an additional finite-imidazole drug called metronidazole on top of this original treatment. After treatment, MG presence was notified, and in the Group 1, MG presence was 14%, while 4% in Group 2. This clinical study suggested that finite-imidazoles may effectively kill MG.
So, what are 5-nitroimidazoles? So here we have 3 individual specific 5-nitroimidazoles that we used and that we studied in our research. And you'll notice a common structural theme among each of them is this nitro group. And the function of that nitro group is when exposed to a reducing agent or reducing conditions, such as our nitroreductase enzyme, it becomes a radical intermediate. And that radical intermediate is highly reactive and goes on to destroy DNA and likely other cellular machinery, killing MG. So the central question of our research was, what are the mutations in 5-nitroimidazole resistant MG strains that might contribute to antibiotic resistance? And we investigated this by sequencing the genome of resistant strains trained against the 3 antibiotics that I mentioned earlier. And by sequencing that genome, and by seeing the mutations in the genomes that those resistant strains develop, we might get clues as to the mechanism of how that antibiotic resistance is developed.
So what was our experimental procedure? Firstly, Victoria Bowman from the University of Washington grew wild-type bacteria, MG, in the presence of these antibiotics at the concentration of 100 micrograms per milliliter. Now, MG's natural susceptibility is below 12.5 micrograms per milliliter, making these samples, which indicates that these samples were truly antibiotic resistant. Also, only the resistant bacteria were isolated, purified, and grown, and such samples were given to us from Dr. Gwenwood's lab. This table summarizes the samples that we received. After receiving the samples, we extracted the genomic DNA using the Lucidin master DNA purification kit. After extraction, we verified that our DNA samples were of high purity and high concentration by the nanodrop. And then, it was imperative that we were working with genomic DNA samples that was of high molecular weight. For this, we ran an agrogel electrophoresis to make sure that we were working with long lengths of DNA fragments. And then after extraction, we processed it by DNA barcoding. DNA barcoding color codes each of the financial imidazole resistant samples so that we could later identify them and separate them within the mixture. And then these dark DNA barcoded samples were then inserted into a benign sequencer. Specifically, these samples were placed in the flow cell, which contained the motor protein and the nanopore. The double-stranded DNA coming from the cis side would enter into the motor protein, and this motor protein has helical function, unwinding the double-stranded DNA into single strand, and these single-stranded DNA would then enter into the nanopore, where into the trans side. And while the nucleotides are flowing through the nanopore, each nucleotide is exposed to an electrical current. And the disruption in that electrical current generates the sequencing data, which was made possible in the Minno software in the laptop.
Yeah, so after sequencing all these strands, we had gigabytes of data to sort through. So we needed to do that through some software. One of the things we had to compensate for first, though, was the fact that when a MinION sequencer has about a 15% error rate for any particular base that you're reading. So we need lots of data we need we need high coverage of the genome and lots of copies of the genome sequence so that we can compensate for that 15% error rate for all of the data. For even our lowest sample, we were able to get about 130 times coverage of the MG genome. To make an analogy, that's if the MG genome was enough letters to fill up a book. We had enough letters or base pairs sequence to fill up a genome or a book 130 times. And if you read through a book 130 times, you'd see the misspellings pretty quickly, pretty clearly. We also had long read length fragments, about 3.5 kilobase on average, median. And this helped us with mapping certainty. So if there was a repetitive sequence, we were able to place it among the 500,000 base pairs of the full genome. So then we used a software called IGV, or an integrated genomic viewer. And that is what we're seeing here on the right. This is a view of the program, a generic view. Up here we have a coverage track, and that is sort of a summary of all the data below. And so in orange here we have examples of like insertions in the sequence or mutations in the sequence that are away from the normal or the wild type sequence. And on the bottom here we have that wild type sequence or the reference genome that we expect in a typical Mycoplasma genitalium.
We then looked to identify which of these mutations were non-synonymous. On the top here in orange is an example of a synonymous mutation where the A to G base nucleotide change does not actually change the codon which is being encoded for. On the bottom is an example of a non-synonymous mutation where the A to C nucleotide change actually did result in an amino acid change. In this case, a change from lysine to asparagine. After going through our whole genome, we identified 7 non-synonymous mutations. One was in MG179, which is a transporter ATPase protein, and the other 6 were found in MG342, which encodes for an FMN reductase. Out of these 7 mutations, there were 3 different types. The first type, shown in red, is a single nucleotide point mutation. In blue is a single nucleotide insertion mutation, and in green is a single nucleotide deletion mutation. The remainder of this protein will be focusing on the MG342 gene, specifically looking at its function.
The presentation.
Yes. We'll be focusing on this MG342 gene, its function, and the mutations associated.
So what does the FMN reductase gene protein do? This slide is a potential model for how this enzyme metabolizes this drug and eventually kills MG. This is an MG cell, and inside it is a circulating DNA. Firstly, the 5-nitroimidazole drug enters into the cell and becomes reduced by the FMNH2 to a toxic nitroradical form, which then binds into the DNA of the MG and leads to mutations and causes DNA damage and eventually cell death. Therefore, this FMN reductase is needed to metabolize this drug, and in order for this drug to be effective in killing MG. Also, but mutations in this FMN reductase may inhibit finite imidazole reduction, which may confer antibiotic resistance in these mutant strains.
Like Avi mentioned, we believe that the mutations in this gene can result in the antibiotic resistance. The first type of mutation, like mentioned, is a single nucleotide change. In this case, it was found in 6 of the samples, and here on the left is an example of a G to T mutation. This mutation altered the ATG methionine start codon to an ATT isoleucine codon, which can alter the translation of the protein. While ATT may act as an alternative start codon, this may not be without a reduction in the protein's expression. The second type of mutation was an insertion. In this case, we see a single T being added to the mutated sequence. This causes a disruption in the reading frame where there's a frame shift and ultimately a preemptive stop codon that results in a truncation of the protein. Similarly, a deletion is when the T was removed, which also caused a frame shift and a preemptive stop codon forming. All these mutations are seen disrupting the translation of this protein, ultimately or potentially impacting its resistance to results.
So we wanted to kind of model how these mutations, especially this frame shift mutant, might lose that functionality of its FMN reductase. So we took the genetic sequence of that reductase, the mutated one and the wild type G37, and converted those to the protein sequence using the known way that MG transcribes its genome. And then we ran that through Google's, very relevant to the talk we just had. Anyway, so we ran it through Google's DeepMind AlphaFold AI protein folding and structural prediction algorithm. And we got these two overlaid structures here. In orange, you'll see the wild type, which is what we expect for MG. And in green, you'll see the frame shift mutant, the M102 sample. And you'll see here that although the structures are really similar, the frame shift, its transcription stops early. It stops right here while the wild type has 2 alpha helices domains and 2 beta sheet domains that remain. And we think that by losing those domains, there might be a loss in function or at least a reduction in function for this FMN reductase.
This study can be concluded in the following aspects. We found mutations in the MG179 and MG342 genes. In this talk specifically, we expanded on the MG342 gene, which encodes for a putative FMN reductase. And we found mutations that may lead to decreased expression or a loss of function, which may allow MG cells to confer antibiotic resistance. More research can be done in the following aspects. We found an ATT alternative start codon in one of the missense mutations. So more research can be done to assess and confirm if this ATT codon may be an alternative start codon. Secondly, it would be interesting to note if this MG342 gene, which encodes for this enzyme, may be this gene mutation may be found in mutants that are grown in vivo. And thirdly, it would be interesting to study the function of those missing domains in the predicted FMN reductase protein model. Lastly, we thank the following people for this research: Dr. Wood and Dr. Nguyen for this opportunity, and also Victoria Bowman for creating those strains, and our colleagues from Molecular Biology Classroom for their collaboration and support, especially Helena, Leanne, and Juan. And this research was possible from this NIH grant.
All right, we've got time for one question, folks. And I can fill that in. And the question I've had for you is, your case is we need more antibiotics for this organism because it's becoming resistant to the one's first line of drugs. Why are you looking at mutations that get you resistance?
Well, one great example is that azithromycin. It has a high success rate between 85 to 95%. However, it's been noted that it's becoming slowly resistant to such antibiotics. And so, because in 2020, fine nitrogen results have been effective in treating MG cells or killing them, it may be that MG may also grow resistance against these fine nitrogen results. And so therefore, it would be prudent to note those antibiotic resistance and those mutations that may confer antibiotic resistance, and therefore to prepare for the future, so to speak.
All right, folks, let's give them another round of